All right. Okay, found the microphone. Thanks, everybody, for coming to this. I am really, really super psyched about this. I think we've been able to do single-cell proteomics for a long time, right? That if you just spend enough time on one single cell, you could do single-cell proteomics, right? You can eventually do it. And it's just really interesting to think that it's maybe went from something that isn't just a toy for mass spectrometers at LCMS to just like show off that they could do it. And maybe we can get to a point where we can make this an approachable technology for biologists. And thank you, ABRF, for letting me put this together. Everybody that came here, thanks for Bruker for helping sponsor this. So may not get the move. All right. No, nope.
Is it on? There's a little button on the side.
Should it be red? Oh, no, it's green now.
Let me just restart your deal share.
Oh, cool. How loud is that? Okay. That's right. Okay, yeah. And so I've got I have a bias and a vested interest in this because I did, go through a couple of rounds of like tenure track interviews, and I certainly had part of my pitch like, sure, we can do single cell proteomics in your core eventually. And it did work out for me. I have a 20% appointment of my effort is into trying to get single cell proteomics working in the core at Pitt over the next 3 years. So I'm going to say yes, it probably is going to work just fine. So we're going to divide this out. Justin's got really beautiful data and these are our speakers. Justin is professor at Iowa State. Oliver's going to show some spatial technology that I haven't seen yet. So I think this would be really cool to just really tie it all together in a spatial context. So I'm going to do the LFQ stuff. I do a lot of multiplexing as well. Most of my stuff is that, but Justin's data is better than mine. So quick overview. I want to say, why would we do single cell proteomics anyway? I'm not going to do this in any order for this like here, but what are the hurdles in 2025? This was going to be a very different list than it was, say, in 2017, 18, 2020. There's a different list now on what the actual hurdles are. Where are the bottlenecks? What are the most approachable technologies? What are the best technologies from a cost recovery standpoint as well, to focus on that, right? And so why single cell proteomics? And I guess, with any single cell technology, you could say, look, we've got these complex cellular environments, we're homogenizing all this stuff together, we're losing all the important stuff, right? Rare cells in a population make a lot of sense here as well, but again, you can say this is for all single cell technologies, right? We're going one cell at a time. We can start to find these rare populations. Most of my work, when I was at Hopkins, all of it was on, pick a cool drug that just came out, let's see what the proteomic response to that drug is, then let's do single cell proteomics and see if the cells are adapting in one way or two ways or three ways to that to try to accelerate chemotherapy, combinatorials. The point where you can step away from all the other single cell technologies and say, we're the only ones who can do this, is the post-translational modifications. And it's really surprising that when you're not homogenizing a whole bunch of cells, that it's actually in the high abundance proteins, it's somewhat easier to see the PTMs in single cells than in a homogenate, which is really surprising, right? And then we tend to break up, diseases into, the genetic diseases and the cancers and things, and then the non-dividing cells that are terminally differentiated that are, those are more protein level diseases, and so terminally differentiated cells are going to have a little more difference in the proteome where the genome is going to be kind of, maybe it doesn't ever change again because it doesn't divide, right? And if you want me to just lie to everybody and cancel everything, because you want to talk about low cell input diagnostics, I will cancel everything. Carl Meschler's got some just gorgeous data where they're sampling, they're doing sampling in people's heads, and they're getting 50 cells or whatever. And if you need a reason to spend, to buy the best instrument in the world and do 10 cells a day, it's one of those things. So any of those things I think are really important here. But again, I mean, really, but in this environment, really, who cares, right? I've been in a couple of quarters, and maybe I'm a little biased by it, but for most times, it's always always kind of this, sorry, Ron, but Dr. Fancy Pants is going to go, hey, he's going to send down somebody to the core and with a nature paper, and my boss is going to say, yeah, we can totally do that, right? And then my job in the core was just like, damage control. And so really this talk is like, what's the approachable way to deliver label-free single-cell proteomics? If you send that to Ron, I will laugh so much if somebody took a picture. So he's actually a nice guy. But so how does single cell? I like label-free proteomics because it's easy to just explain how single cell works here. There's magic, the facts or whatever it is, and all these different technologies that I don't know how they work, and I'm starting to learn how they do. But what I need is one single cell to end up in the bottom of a tiny well, as small as possible. I need that to land there, and I need that to be intact when it goes, because if you've got a busted membrane, I don't care about the proteins there, right? They're going to leak out, and so that's a big step aside from some of the genomic stuff where the cells can be lysed. They can come from different places where you really are just looking at the nucleus, right? And people do this in different ways, but essentially what you need to do is just lyse that cell once it gets there, and then use trypsin to chop things up into nice little pieces. Thank you, BioRender. And then, while you have your peptides out, you just need to just load every single bit of it into a mass spectrometry system. And I use this vendor's instruments, and maybe Justin uses this vendor's instruments, but at the end of the day, it really doesn't matter. If you're patient enough and you've got a high-res, well, you don't even need a high-res system, you need a trapping system that can accumulate ion signal. It doesn't really matter. You can do single-cell proteomics. It's can you do it in a reasonable time frame, right? So just this quick aside here. So for anybody that's not in mass spec, right, we take the 3D proteins that are a mess, we reduce those to linear chains. We like trypsin because it's super, super efficient, and it chops the peptides, statistically every 13 amino acids, right? The peptides that come off, the second best thing about trypsin is that it leaves lysines and arginines on one side. At acidic pH, that's always going to charge, and then pH, pK stuff at pH of 3, you're basically going to pick up a secondary charge somewhere on that sequence. We simplify that because the mass spec can't look at everything at once. We do HPLC, it will never look like that. But as the ions, as the peptides elute off the liquid into, they come right off the instrument almost always now, where they come off onto a little needle where you charge it with just a crap load of direct current, maybe some heat to convert that to gas, and then that gas goes in the mass spectrometer. You accumulate all of the signal that you can for your ions to get your masses, or actually your mass per charge ratio. Then you again accumulate your target ions, whether it's the most intense or whether it's just this packet that have this particular characteristic. You accumulate those, you fragment them. You probably don't use sequest, but that's the original one. But you take the fragment ions and your parent mass and you match it to a database and you say, hey, this is what that peptide is, right? And so the difference in single cell proteomics from label-free is that you just use a ton of accumulation time. And so when you look at some of the early papers that were done on, like the Orbitrap fusion systems, like 1 and 2s, or well, I don't think it was a 2, but a 2 and 3 systems, you'll see they'll use like 500 milliseconds of fill time, right? So they're accumulating for almost a second. And you think like, that's not a very fast sequencing thing if you're only sequencing 2 peptides a second, right? But you can just crank up that accumulation time and you can get that signal, right? And so that illustration, this is probably just one of the best examples of this from a label-free perspective. It's exactly what I just told you. So this is out of Carl's lab, and they just sort cells in a 384 well plate. They prep in 1 microliter total with a nice surfactant. An Exploris 480, which is a fantastic instrument. It's a few years old now, with FAMES, running 30 cells a day. Can get you. And I have reprocessed all their data. You can get 1,000 proteins in the easiest human cell to do proteomics on. And if you do match between runs where you're saying, okay, well, this peptide ID, I don't need sequence coverage of it. I'll just say if it's got this mass and retention time, that's it. You can push that up to 1,500, 1,800 proteins. They do use an expensive picoliter handling robotic system for this paper. There's no reason that you needed to do that because you just don't have to for this workflow. They're just very good at using that expensive robot, so they just used it there. And I do know this Exploris 480 personally, and I think it probably was dedicated to single-cell proteomics for its entire lifespan. So it probably never did anything. So could you have an Exploris in your core that did plasma this week and that did single cell proteomics next week, yes. Could it get 1,000 proteins per HeLa with the exact same method? I don't know, right? Because, is there a benefit to having a system that has to be, dedicated to ultra low input, right? And so I think a lot of people know Brett Finney, who's run one of the, the longest historic proteomics cores in the U.S. A fantastic person. Brett has said the whole time he's going to wait till the dust settles and everybody stops and decides on a method, and then he will start doing single cell proteomics. And so it's been fun, that as we've been working out these bugs over the last 6 or 7 years in single cell proteomics, there have been some surprises, like wide window acquisition for data dependent acquisition works really, really well in single cells. And we think that's because if you're looking at the log copy number distribution of all the HeLa proteins that you can see from like 10 copies down the transcription factors of the histones at 10 million copies, this is big, wide range, but the blue stuff is what you actually see in single cells. So you're only seeing the high abundance proteins. So if you've got a great big window and there's only 1,000 detectable proteins there, you're not as penalized by having more ions fragmented because it's just this stochastic thing. Surfactants just popped up that I'd never heard of before, and they can just about double your IDs. Tom Gal's got a beautiful paper where he goes through and just picks different 384 well plates and shows you get like 20% more IDs by using this plate versus that plate because it's a different plastic. So there's things like that have just really allowed this field to get better. And then Orbitraps do some interesting rounding stuff at really low levels. And if you adjust your search engines for that, then you can get a lot more out of your data. So a lot of surprises. So in a core, we've always got to manage expectations, though, right? And proteomics people just fucking love counting proteins. They just count proteins, like, I only got 15,000 proteins. And this has not stopped at SCP. And these assholes right here put in their preprint title, we can get 5,000 proteins in a cell. Right? And some of these are my friends, but it's like, I hate this paper inside and out. But yeah, you absolutely can, if you buy an Orbitrap Astral and you buy a picoliter robotic system and you spend all day on one cell, you can get 3,500 proteins. I don't know where they got the 5,000. But the picoliter robotics are slow, they're expensive, their consumables are expensive, long gradients are expensive, right? Patience is expensive, right? And so just thinking about this from somebody that has been a small business owner on the side for running mass specs, I think about this stuff from a cost perspective, and a mass spec's a lot of money, right? And Hopkins calculates it depreciates to 0 in 7 years, and if you don't believe that that's real, try trading in last generation's Orbitrap on this generation's Orbitrap, and they might take it away for free, right? And so if you assume, just in a stupid, just crazy idea, right, that you really just wanted to get more proteins than Olsen Lab, and you just ran one cell per day, and you did that for 7 years until that instrument depreciated to $0, you get 2,500 cells in that 7 years. And so that would cost you just in the instrument itself, not $200 to $600 per cell, right? And that's a stupid example, right? Nobody's running one cell a day. A lot of people are running 10 cells a day. So you divide that by 10, and you're still at $20 to $60 a cell, assuming no labor, no service fees, no downtime, and no reagent costs, right? And I can, there's a group in Gaithersburg, I can take them a vial of cells with a 2-condition setup, and they can do 12,000 cells for single cell seq for 5K, and so that's 42 cents a cell. That might have been a Christmas deal, but I'm holding on to the quote, so they have to still do it. And there's other costs, right? So how you sort the cells can be really expensive. So FACS in the core at Hopkins, 120 dollars an hour on a MoFlow sorter. We did 8-condition study, 4,000 single cells were sorted. That cost me $240. Got me to cover this nitpicky nerd journal. And but if you go to the picoliter robotics, the Cellum one, it's an expensive instrument. I was code 350. if you're doing it with the way that Olson Lab is doing it with them, where it only preps 96 cells that day, that's what it can do. It still requires $10 in reagents per cell to do that, right? So that really just cranks up how much this costs. The other consumables, if you spend time aliquoting them and things, there's virtually nothing to it, right? 2 nanograms of trypsin per cell is what I use. I can aliquot that out. I certainly don't aliquot it out to perfect efficiency, but that's a lot of cells. One TMT 16-plex kit could do 60,000 cells. I'm not gonna do the math for the new kits. But then, Evo tips, I'm using those. I like the Evo step a lot. I hate nano LCs, but that one's okay. It's $2 a tip, right? So I have to think about those things, right? And so, and probably one of the least popular things I've ever written, Amanda Smithers and I went back through all the single cell proteomics studies, and we backtracked and we tried to figure out how much each cell cost. And so you'll see that there's this great big range, right, where some of the multiplexing stuff early in and Justin's stuff, right, that's coming in at our calculated cost of like $1 a cell, right? Yeah. And not to pick on in this study, because this study of Ryan Kelly's lab is maybe one of my favorite papers I've ever read. They laser capture microdissected neurons out of healthy brains and brains with ALS. They only did 9 cells a day, right? And at that throughput, right, it ends up being a $55 cell study. Totally worth it for that study. It's fantastic, but you got to think about those costs. And so, so here's LFQ workflow number 2. This is just the example that I'm using. So I'm using a TK Uno single cell printer. I was the first person who had to pay money for it. They were just giving it out to everybody, but I got to buy it. I got over $25,000. The reagents are now at $50 a chip or whatever, and it'll print a 384 well plate, but it won't print 496 well plates. You got to change the chip because you take it off and it's like, gotcha. All right, so we got to figure that one out. But this is my current prep. We deposit the cells directly into an Evo tip. If you haven't seen an Evo tip, it's just a P100 pipette tip with a tiny amount of C18 in the bottom. We put the lysis buffer and trypsin on the top. The TCAN drops the cells in. I incubate for more than 4 hours at 37. The paper says 1 to 4. Don't do that. It's more than 4 hours. You quick wash the peptides after they're digested. They stick to it. You load it on it. I'm currently doing 40 cells a day, but I'm also a new PI during a hiring freeze, and so 40 makes a lot of sense for me. I can prep cells on Friday, and then the instrument can run for 2 weeks, right? My students had a lot of trouble with this early in, and I don't know if it was a software issue with the TCAN, but we were getting on the timsTOF Flex, which was essentially a Pro 2 model, about 500 proteins per cell. Our SCP was getting about 850, and yeah, I don't know what the Ultra is. It was just installed last week, but I'm expecting much bigger numbers out of it. Oh yeah, and this is it printing, right? And so it's just depositing the bad cell, it moves, it deposits the cell, right? Yeah, no, and I'm not using any bioinformatics, right? This is actual peptide IDs. It's not match between runs. It's not a distorted library. These are real numbers you can get with this because it's really so low loss. I didn't come up with this. Yeah, they beat us to press with it. They called it one tip. We were going to call it Unitip, and yeah, their data was better actually, right? But so my costs, right? So right now, because I can only print 96. One box of Evo tips for one chip is about $5. The tips cost me $2. If I do the whole math I did before, 14,000 cells a year, timsTOF Ultra 2 is probably 1.1 million, right, with an Evo step. You throw in the service cost, you think about that. So you back calculate that. I'm running about $22 a cell labor, which is a whole lot more than I was hoping to do, and for way more than you do with multiplexing. But we know multiplexing is cheaper. But I rent space off the phenomenal Pitt Health Sciences mass spec core, and I took this from their page for what they charge internally. So if you think like, hey, I'm getting 2 cells in about an hour for $44 to prep it, somewhere in there it kind of makes sense, right? So this is probably not an insane model for studies where we can get away with just a few cells. And for the big studies, I think you really, if you want to do a big study, you got to multiplex. And so just, I 9-plex multiplex. I use 10 TMT channels on the timsTOF. Yeah, and so after controls, I get 7 cells per injection. This paper will be out soon, but I tried to push the Evo step to see how far we could go. Yeah, you cannot do 5. If you want to poorly measure 3 histones, you can do 3,500 cells a day. But 30 speed, because 210 cells a day, it looks just fine. 420 cells or 700 cells are just great. I can still see histone PTMs pretty consistently at 700 cells a day. And so that's, if I need to do a big study, that's how you do it, right? The current challenges are still getting that cell isolated. 7 out of 10 of my collaborations, they find out like, oh, they actually already killed that material. It was sliced and fixed, and there's poison in it, and that cell is definitely dead, and they can't sort it, right? Or other things like that. They can't sort the cell out. The data analysis, this is where we start to say, hey, this is really hard, but I mean, just getting to cell is in this column, protein is in this column, and protein abundance in this cell is, that overlap. That's not hard to do, right? And if that's, interpreting past that's tough, but if your deliverable in a core is a sheet that's like, this is the protein abundance in this cell, this is a functional deliverable. It's not that tough to do, right? Quick takeaway, SEP is way better than I think the last time I was at ABRF, it was 5 years ago, and somebody spoke here, and it was more of a like cool science experiment they can do at Northeastern. But your costs, there's a lot to think about costs, and how you prep your cells is where a lot of it can come from. I think that there's a point where a lot of this, I think that we're going to start to see these offerings soon. And if you do take this route, like you decide to do this, there has been no better investment of my time than sitting down with a fax person and like finding out whatever the stuff is that they know and make sure that they understand what we're doing, because they're actually serious scientists as well. And I feel like a whole lot of the failures in single-cell Zeek is like we just dropped stuff off at a core and we didn't talk to the people that did that work. That really, that's where my studies have worked, is when we've sat down with the core people up front. Quick acknowledgements, thanks for the money. Ahmed and Colton, they kind of got the TCAN thing working. But yeah, thanks for that, guys. And then that was it for me. So we're gonna try to hold the questions till the end, and then we'll all be up here and you can just ask us stuff. But but Justin Walley is gonna go next.
Okay. So first, I'll get started. Thanks, Ben, for the invitation. It's my first time at this meeting and I've been enjoying it. So my lab is a plant biology lab. That's what we study. We're not a core facility. I joked last night with Ben, we're soft core. So we have mass specs and we collaborate a lot. So I'm kind of like a drug dealer where I get people hooked and then I get to end up on grants with them. But I think of ways that we can do interesting science and do it in a high throughput manner and deliver lots of data to folks. So again, I'm at Iowa State University. I'm a plant biologist. So I thought I'd do a couple slides to kind of highlight where I'm coming from and wanting to do single cell proteomics. So we, specialize in mass spectrometry where we're measuring protein abundance in PTMs. We also measure transcript levels. And we do this in different contexts to understand how plants perceive and respond to the environment to optimize their growth. And we like generating lots of data and then reconstructing networks, whether they're transcriptional or kinase signaling networks, and then use those to get at actual biology where we do kind of high throughput phenotyping of mutants and different things we find that are important in these networks. We go from these large scale networks down to specific molecular mechanisms. To do this, we rely on generating lots of data. And then thinking to switch to single cell proteomics, I've always thought of how can we analyze lots of samples and get data of interest. And then this context, Ben highlighted this with single cell RNA sequencing. This is 6 years ago now. I'm going to talk about roots later on, so I just pulled some single cell root examples. From 5 to 10,000 cells, they're already sequencing the transcriptomes of those, but you're able to identify across roots all the major cell types. And then fast forward a couple of years, we're at now hundreds of thousands of cells being measured in different single cell RNA sequencing experiments. And now you can not only get the cell types, but kind of how they're behaving over time. And this, got me thinking when I started on developing these or trying to develop or optimize these for plants, is that the goal really should be to get to hundreds of thousands of cells being measured for single-cell proteomics. And that's, still not achievable, I don't think, in any real meaningful way. But, really you can get super cool biology from only a couple hundred cells being measured, and I'll kind of highlight that. But it's this idea that we need to do methods and develop methods that scale for lots of cells. And this kind of gets at this bottleneck, which is the throughput for single-cell proteomics. And you can do very nice experiments, as Ben highlighted, with label-free proteomics, whether it's DDA or DIA. And then how can you increase the number of cells you're analyzing in a day? You can multiplex with either tandem S tags, which works nicely, and I'll kind of focus on this. And this idea of multiplex DIA is really intriguing, and hopefully it kind of takes off. We're still waiting on the reagents. And I'm literally waiting on the reagents. I ordered M-Track 2 years ago, and it's still back ordered. But the idea here is, label-free. With the current plexing for DIA, you can do to 3 to 5 cell plexes. And with TMT, you can now get up to 32 plexes. So you're getting in, depending on your instrument time, 100 to 1,000 cells a day now with these multiplexing strategies. And there's lots of ways to handle the samples and get proteins into a well and analyze them. Ben highlighted there's a nice picoliter robot that's expensive, and I kind of break it down as to methods that require that robot and methods that don't, and we don't have that robot, and I don't think most cores will have this anytime soon unless they're dedicated to just doing single-cell proteomics. So we've developed and worked with these MPOP methods, which are essentially what Ben was describing minus the TCAN Evo tips. And this multiplexing method we're using was developed by Nikolai Slavlov's lab called Scope 2. And the idea here is you use tandem S tags to label and multiplex samples. And in these tags, we're working with the TMT 18-plex versions where you can label, each individual single cell. Well, first you isolate them, digest them, and then you barcode them. But you end up barcoding a single cell. In the scope context, we usually use a reference of around 5 cell equivalents. So this helps normalize across multiplexes. When you're doing lots of multiplexes, you need to kind of normalize the intensity. And then you can add a carrier to help boost identifications. And that helps, again, boost identifications. But then you get your barcoded peptides. You get your MS2 where you have your carrier, your reference, and then your single cell being quantified. And then Nikolai's lab and others have some nice packages where this DoMS is nice for optimizing your acquisition settings. And then when you're actually acquiring data, we use their Dart ID and then a SCP package from another group, which I'll highlight in a minute. But the data analysis workflows are kind of worked out and nice to use. And a couple points along here is this reference and carrier, it's a fair number of cells. Often this is done by depositing with FACS or the Selenium 1 actual cells into a well. Plant samples are well known for having lots of secondary metabolites and fouling everything that they touch. So to avoid this, we actually used our typical bulk proteomics sample prep method and then aliquoted peptide equivalents for 5 referent cells and the number of carriers we end up using. So again, it's actual purified peptides that were used to label rather than isolated cells. And then another thing to highlight is this carrier amount is very mass analyzer dependent. So there are many papers out there describing the optimal carrier and how everybody else's carrier is wrong. And what it really is depending on the mass analyzer you use, it can handle more or less ions at a time. Often in this, it's recommended to omit certain TMT Pro labels because they can have isotopic carryover. I'm not sure if this is still true, but we did avoid those. Here's a number of citations for this workflow that's the basis of it. And the 2 Nature protocol papers are really nice, outlining how much to put in a well and how to move it around. This DoMS again is nice for kind of optimizing acquisition and settings. It gives you a number of parameters for ions you've counted and how pure they are. And then this SCP package I'll come back to in a bit. So when we started out, we wanted to know, one, is this feasible, and what all do we need to change from what we're normally doing? The math that's the basis for how much we're inputting here is we measured Arabidopsis root cells, and on average, they have 170 picograms per cell. They vary. Their cells are different sizes and shapes, and I'll also come back to that. But if you take in, you're going to use a 5-cell reference, 14 single cells, and then we tested 100 or 200 carrier cells. That works out to about 20 to 37 nanograms per multiplex set. So we tested different input amounts. We tested the Slavlov Labs optimized SCP settings. So they used a Q-Exactive, which is similar to the Q-Exactive Plus that we used. And for fun, we tested our typical bulk acquisition settings for tandem S tags. And then here's a few, but we changed ion injection time and AGC and all that and then tested it. We also tested typical columns, so we typically use this Pharmafluidics, now Thermo micropack column, so it was the 200 centimeter version from Pharmafluidics at the time. Surprisingly, I'll come back to that, but it worked better than the 50. I was not quite expecting that. And then we changed different LC gradient settings. And to give a highlight, I'm not going to show all the acquisition tests, but just to show if a standard bulk TMT acquisition you use versus an optimized for single cell, you can see, oops, my animation's not working, but you about double your identifications, whether it's proteins ID'd or quantified, and similar benefits are seen in terms of ion purity of what you're actually quantifying. So again, we ended up, using this settings from the Slavlov's lab. We shot for 37 nanograms. It worked well. We used a 200 centimeter micropack column, a non-linear gradient that we switched from our typical bulk proteomics gradient. I think we used Gradient Optimizer, which I probably found years ago from Ben's blog. And then a 110-minute gradient at 300 nanoliters per minute. So again, we tested these. It's now at the point where for the mass spec and LC you have, there's probably already optimized acquisition settings out there that if you use those, you'd be good to go for, I think, all the major mass specs on the market right now. So with that, I'm going to talk about the actual study we did, and this was doing single-cell proteomics on 2 cell types from roots, the cortex and the endodermis. This was done by Christian in my lab, and then Trevor Nolan, who was a postdoc at Duke at the time and is now a professor at Caltech. But to kind of orient you on roots, so the roots grow. They germinated from a seed here, and they grow from the tip. So all the growth is happening at the bottom of it. And what's happening at the bottom of that tip is there's a root apical meristem, and that meristem contains a population of dedifferentiated stem cells that ultimately differentiate into all the cell types in the root. So you have the stem cell population, they differentiate. Another feature of this is, so this is where the stem cells are. In this meristem proliferation zone, cells divide. And you can see this is color-coded here, where each new cell that's divided and gets a color code, and you can see new colors popping up along here. And then above this line, no new colors appear. Cells are rather now just expanding. So you have cells dividing here and cells expanding and getting bigger beyond that zone. And what this ends up providing is this developmental timeline along the whole gradient of the root. So this would be the tip, the new cells. This would be near where it germinated at the top. So there's a longitudinal axis, has all the developmental stages of the root. And then similarly, if you go latitudinally in a cross section, all the major cell types are present in the root in these concentric rings. So you can get both space and time using this root system of that. And then the specific cells that we focused on are from the cortex and the endodermis. And these share the feature of they differentiate from a common stem cell. So the stem cell at the tip of the root divides and differentiates either into the endodermis or the cortex. And so we're having these 2 cell types coming from the same stem cell. They're adjacent to each other in the root. So they're very close in developmental staging and proximity to one another. And we wanted to ask the simple question of, could we use single-cell proteomics and actually tell that these two are 2 different cell types? Or are we just simply measuring a bunch of stuff that's not different between different types of cells. So then our workflow, we had marker. The other nice thing about Arabidopsis roots is there's fluorescent tagged marker lines that mark each one of those cell types. So Trevor sorted the marker lines for endodermis and cortex using FACS, deposited them into 384 well plates, and then we performed that in-well lysis and trypsin digestion and TMT labeling using an Opentrons OT2, and ended up again, we ran out of carrier, so we ended up having to divide and only use 135x carrier rather than the optimized 200, but still worked well. 5-cell reference, and then the single cells. And I thought I'd show how we're actually using this Opentrons OT2 for this. It's turned out to be a nice system. Christian made the video for this last week, but I think I hit the wrong button. So we take frozen plates that were shipped to us, put them at 90 C to lyse the cells. You can cool the plate down. We're adding trypsin benzonase solution. At the time, DDM wasn't on the scene, so we didn't use it, but I would if I was doing it now. And it'll go through, and we keep foil on top of the plate so they don't dry out, but the tips puncture really nicely. We put fresh foil in after adding the trypsin. We're adding the TMT Pro label now, and this magnetic module was used just to raise the tube up so it didn't have as long as transfer times. We just had an Opentrons Flex installed, and now these modules are in the deck, so there's not as much of a height difference. And the other nice thing with the Opentrons is you can put in a pre-programmed list and randomize where cells are going to go when you pull them, so it'll go through. I couldn't do that by hand and not mess everything up, but so you can randomize your labeling, you can randomize your pooling, and you can pick the different labeled cells now and put them into your auto sampler vial. So it's a nice system for tracking and randomizing as well. And the other system that Ben highlighted was the TCAN Uno. So we're hoping to set this up, and I thought maybe we'd have data, but don't quite have it yet, where if we can use this also for a scope style multiplexing and sample handling. The benefits are there. It's nanoliter now instead of microliter, which is the Opentrons scale applications. But I wanted to highlight that both the Opentrons and TCAN are nice because in a core setting or any other setting, they're not proteomics specific. If you're sharing them with the genomics lab, they can use all these. The TCAN comes pre-programmed with qPCR assays, for example, and the Opentrons obviously is used for all kinds of things. So you don't need to buy a dedicated instrument just for this one application. And then, we acquired this data, and coming back to this SCP package, has a nice sensitivity analysis, and then this paper in Journal of Proteome Research documents why these kind of QC checks are nice to use. But, you get these nice graphs and things like cumulative sensitivity. So in here, every time you add a new cell, you see if you find a new protein, and you can see we saturate after about 100 cells. We don't detect any new proteins. It gives you your local sensitivity, so we're just over 1,000 proteins per cell. It gives you a measure of data completeness, so this is the sensitivity, so how many you're measuring on average per cell. And I don't quite understand the scaling of this, but it gives you a scaling of how often there's a missing value. And it gives things like Jacquard index, which shows consistent protein detection within cell types, and it's kind of a measure of the overlap in features between any two set of cells. But this provides a couple of nice metrics. One, we can say, did we mess up one of the cell types, and we're going to find differences because we have different QC metrics. And you can also preload lots of other groups, single cell proteomics data to see if you're kind of in the same ballpark as other studies have been. So that's a nice paper I wanted to highlight, and it's a very good bioconductor package. What we ended up doing is measuring proteins from 768 cells. Did a number of filtering steps. So for identifications, we're at about 3,700 proteins, 1,100 per cell. And we added a number, and a number of the groups have used this different filtering steps, so coefficient of variation and missing peptides and stuff. And you can see once we add some of these filters, we drop down, and this coefficient of variation filter was important because it actually had us eliminating a number of the cells we measured. So we ended up actually focusing on 81 of those 768 cells just because of this variation. And I'll come back to why I think that's biological. And the other point from this cell, or this two other points, we ended up with about 596 differentially expressed proteins between the cell types. But this was acquired on a Q Exactive Plus that came out somewhere, that model came out like 2013 or something, and this instrument saw more of the Thermo Service Engineer that year than it did samples, so it wasn't like the best running mass spec you're ever gonna find. And some benchmarking for what we found. So we went back the same, Trevor was in this lab from the group that did the single cell RNA sequencing paper I mentioned. So the plants were grown in the same conditions and handled the same way and sorted on the same sorter. So we took their single cell data, transcript data, and a rank ordered transcript abundance from low to high and low to high. And you can see I've transcript abundance across this whole range, but we color-coded the cells in red that we detected with proteomics, and you can see all the way from high abundance transcripts to low abundance, we're detecting protein, so we're getting a pretty big range. There is a bias for detecting higher abundance transcripts, so these ones were detected by transcriptome and proteome. You can see a number of genes over here have transcripts that we can detect, but we didn't see protein for. But again, we're getting a nice range of genes abundance being detected. We often get asked, can you find interesting stuff like kinases and transcription factors? So from those 3,200 quantified proteins, we have about 175 kinases and a similar number of transcription factors. In here, we're just a similar plot, but now it's the protein abundance ranked two ways. And you can see the transcription factors and kinases span the abundance of the proteins we detected. And importantly, can we find meaningful stuff that's differentiated between or distinguished between these two cell types? So here's a MA plot for average intensity and fold change. Stuff up here is enriched in the cortex. Stuff down here is enriched in the endodermis. And we found a number of proteins. So this transcription factor, auxin response factor 5, was enriched in cortical cells. It's known that this transcription factor drives cortical cell division. Similarly, there's other markers for cortex that we found. And then on the other side for endodermis, there's this feature of endodermal cells is they make this Casparian strip. It's this suberized barrier that prevents water and nutrients from exchanging randomly with the environment. And this Cas1 protein drives the formation of this strip, and it was enriched in the right cell type. A number of proteins, whether they're related to reactive oxygen signaling or hormone signaling, in this case auxin transport control, which is a plant hormone. And then other things we see in here, there's a well-characterized receptor kinase, and SNRK proteins involved in sugar sensing, a number of other transcription factors that are known to drive root development. So we're finding, kind of regulatory proteins that are of interest and in the right place. And then we ask the final question, can we differentiate these two really closely related cell types? We did that a few ways, whether it's principal component analysis or UMAP. You can see the cells from the different types are separated. Similarly, with hierarchical clustering by sample, you can see coded down here endodermis and cortex. By and large, endodermal cells cluster with each other, and similarly for cortical cells. So I think we did show that we can distinguish these two related cell types. And that leads me to looking forward. So there was variability in the quantification I mentioned with the coefficient of variation, and I think this is a lot biological. So when I showed that video, you notice the cells get very different in size as you go along that gradient. So there's cell size differences. There's also cell cycle differences within the root, and I think both of those are part of that quantification issue. And there's a few animal studies that have shown that if you normalize with histone proteins, it helps control for those cell size differences. The problem with plants is the genomes also undergo endoreduplication. So as they get older, the genomes duplicate within a given cell. So the histones aren't a great marker for protein content in this context. So now, on campus, we have a new BD SFAX sorter of some sort. I forget the exact acronym, but it can not only, sort by fluorescence, it can bin loosely by cell size, and also take an image of the cell as it's passing through the instrument. So we're going to go back now and collect cells where we kind of bin them by size and image for the actual size and see if we can find proteins to help normalize. Methods, and then looking forward, methods for samples are processing rapidly. The Selenium one's come up a few times, and I think I've never actually used one, but it looks like if you have the money, it does do really nice stuff. I think this TCAN is a very nice system that can help, and there's more and more custom, things that would be really nice to have, but you won't have them unless they get commercialized. And then, Ben highlighted that the newer coverage from the newer instruments is really changing things, and he's lucky with a nice Bruker instrument. Thermo has some nice ones. And you can get now 3,000 to 5,000 proteins per cell. This is all label-free. And I think this highlights a very important concept. Different cell types have very different protein content, and when you see very different numbers between different vendors, it's often they measure different cells and that are different protein content. And then opportunities for single-cell proteomics. I think the big one that's very achievable is how do individual cells respond to a stimuli or a drug. I'd love to see some single nuclei proteomics, and then moving forward, organ level and organism level single-cell proteomic data sets, and then pairing this with RNA sequencing and being able to predict different regulatory network structures from the data. But all in all, going through this, I think I'd be very optimistic about this in a core setting. The sample handling stuff is not needed. You don't need application-specific sample handling stuff. The methods are pretty robust out there now. And on the reagent side, Ben highlighted the cost is almost negligible compared to normal proteomics. And so with that, I'll thank a few people. Christian did the work in my group. Trevor did this when he was a postdoc with Philip Bentvie. And then Iowa State Plant Sciences Institute has really helped with funding for these exploratory projects. And then the College of Ag and VPR helped me with nice instruments. So with that, thank you. Questions at the end, right?
So when you're looking at the sequencing of proteins, how does this compare to doing something like MALDI imaging, where it seems like you could have a higher throughput with something like that perhaps, but I don't, the ability of the technology and where it's placed is how this is compared to the two differences. I guess just, I don't know, we're all discussing that.
You probably have better.
Yeah, so the cell size stuff, if you use a slug neuron, that's the size reporter, the MALDI imaging works okay. So there's you just don't have it, MALDI imaging looks like Albert, you can load up. All right, is our 5 o'clock? Let me just put my stuff away.
Okay, thank you all for being here, and thanks for the invitation, or I guess maybe we sponsored this and that's why I'm here. I don't know, I didn't pay attention, but here I am. I just took notes here, and I forgot what I wrote down. I need this for my introduction. So just like Justin, I'm a biologist at heart, but my day job's to build platforms, and I built optical imaging platforms. So essentially, I make microscopes, and what I'm going to talk to you today about we call spatial proteomics. I don't know if it's the right thing to call it. Essentially what it is multiplex immunofluorescence. If you may or may not know, immunofluorescence has been around for decades. It's labeling your target protein with an antibody, and the antibody has a fluorophore on it. You take a microscope, you take a picture of that with a microscope. So how does this technology fit into this particular symposium? I wrote down that Justin said that there are 596 proteins in the endodermis versus the cortex, and he shows a scatter plot, and there's 20 or 25 of those he actually annotated. In my mind, 20 or 25 proteins is all anyone will ever study in their career comprehensively. If you study any protein, every protein, good, enjoy, but at the end of the day, we drug one target, or one gene becomes a man's obsession, or whatever. This is how it works in science. So that's where I come in. Once these 590 proteins have been identified, and someone actually only cares about 10, 20, 30, 40 of them, the questions start to develop. What do these proteins do in situ? What is, you know, it's good to know a protein's there, it's good to know a cell is there, but we know, especially from cancer biology today, that the sheer, you know, presence of a cell does not predict disease outcomes, for example. It's important to know, you know, what type of neighborhood a cell exists in, or what, you know, what, of course, the cellular identity, but how do they interact with each other? And cancer is a beautiful example of that. So unlike a plant, in cancer, any cell can be next to any other one in some sense. The stratifications of cancers is very poor. So for us, it's really, really important at a single cell level to see every single cell and then understand, okay, what do these 10 proteins do? That's what we do. That's, to me, what single-cell spatial biology is. So my data look like this. They're essentially pretty pictures. This is a breast cancer slice. It's 1 millimeter here on the bottom. You can see the scale bar if you're in the right place. There's about, I would say, about 450,000 cells on this. So that's how many you get in one shot. This is a couple hours worth of imaging. These are the epitopes that we labeled. So these are the targets that we look at, mostly immune markers that are interested in cancer. And what we do is we take an algorithm that finds every single cell based on the shape of its nucleus or its membrane, draws a little circle around it, and the cell then becomes an object. And then we can measure the fluorescence intensity of any one of these markers within that cell, and pretty similar to what was shown before, we sort cells based on phenotypes, based on what markers they express. And then we can go in and we can use morphological metrics like what's called spatial neighborhoods, not only to quantify what cells are there, or frankly where they are, but how they interact with each other. This is, in some sense, a quasi-functional view down to single cell level of what's going on. Again, it's 500,000 cells here. It's one slice. So this is really becoming something, and a lot of us are heavily involved in this. And you can see there are a lot of exhibitors here that are spatial biology companies. So I would say since 2017, these methods have become in vogue, especially in cancer biology. And again, in the immune system, it's really important to know, you know, what cell is next to each other, because in the immune system, a lot of things are still tactical. It's touching, one cell touches another, activates a checkpoint. This is all local signaling. So it's very, very important to look at the spatial context. So how does this work and how do we do it? This is the machine that we're building in my lab, or it's already built. It's commercially available. Go see the Brooker booth if you want to learn more about it. Essentially what it is, this is like an oversized desktop printer. It's an epifluorescence microscope that lives inside here. There's also a fluidics unit that sits next to it and connects to a computer and all. You take a histology slide, so this would be a tissue sample. It's mounted on a glass slide, and you put this cover slip on it. The cover slip turns that tissue environment into a flow chamber, and what we can do with that is we can deliver antibodies onto the tissue. So basically you put that cover slip on, you put it on a microscope, you close the lid and you go home, actually, press go. Then what happens is you stain the tissue with antibodies. In our case, we use fluorophore conjugated antibodies preferably because it makes for an easier experiment. I don't have a lot of patience for complicated experiments, so this is why I do this kind of stuff. And then we take a picture. This is essentially what we do is we take a series of pictures to make sure we capture the full dynamic range of the fluorophore signal that is there. And then we erase this fluorophore. So fluorophores are sensitive to light. They both lend us the light, the emission to take the picture, but if you shine a light too long onto a fluorophore, it actually breaks down. And in doing that, the tissue that was previously colored and that we photographed has now returned to an uncolored state. So you can restain it, and then you take another picture and you go around and around. So you do this for about five biomarkers per cycle, 4 to 5, until you have, I think most people will do this for about 20 biomarkers, 30, 40, but we've done as many as 120, 130. I don't know what the maximum is. I suspect, so we do it on the nanostring platforms, we do 540 routinely. It's a different experimental approach, but before we get antibody crowding, we should easily be doing north of 500. So it's doable, it's feasible, but the more image or the more colors the more antibodies you put on there, the more time it takes. I'm more focused on throughput, getting more tissues imaged at a faster clip. That's my job. And then, of course, you analyze your data. So now again, you go outside, there's five companies that have a similar product. It's important to talk about what makes these products different. And if you look at spatial proteomics or multiplex immunofluorescence, call it what you want, there are three main approaches that have sort of survived the last 10 years. One is indirect immunofluorescence. It's probably the oldest and most well-established, however, mainly used for low-plex applications, 2, 3, maybe 4 antibodies. However, technologies have been developed to do higher plexity, so doing up to 40 antibodies. And what they do is they actually elute or strip or somehow remove the whole antibody complex after they take a picture of 3 to 4 colors. Then they remove this whole thing, and then they restain it again. And it works for many applications. It's pretty good because it gives you a lot of customization. Oligo tagging has really become very widespread now, especially for high-plex applications. What you do here is you take your antibody, you label it with an oligo, and then your probes, your fluorophores, they come in with a complementary oligo. So it's pretty quick, and it's great to do high-plex with. Problem here is that the oligos can really mess up the behavior of an antibody. So you have to be careful with that, and you have to know your chemistry. There's a lot of poor chemistry out there. We use primarily direct IF. So there are thousands of antibodies already available from vendors that already have a fluorophore on them. These chemistries have been around for 30, maybe 40 years, and the catalogs are massive. So this is a very, very simple way to build an assay, and that's kind of what most of my talk here is focused on. Now, the problem with the technology of using direct IF, you have a fluorophore on your antibody. It's great to take a picture, but to get rid of that fluorophore signal is not trivial. Yeah, you can strip it, you can heat up your tissue, you can put acid on it. There's many ways to do it. We have in the past used photo bleaching. So we use a light, we shine it at the fluorophore, and then the signal of the fluorophore decays. I'll show you that. However, in the past, it's been very inefficient. You could sometimes spend minutes trying to decay a fluorophore signal. For someone like me who wants to do an experiment, like not just one slice, but 20, 30, 40, 50 slices a day with 100 proteins, that's not fast enough. So we recently developed in our lab what's called Epic IF. This is really a game changer, I think. If we would have known about this stuff 10 years ago, I think a lot would have gone different in the spatial market. Epic IF, I would say, was a somewhat lucky discovery in the lab. But what it allows us to do, it allows us to zap any organic fluorophore, which is great, because now you can go into an antibody catalog. It doesn't matter if it's an Alexa fluorocorrelate, dilite, whatever color, you can work with it. Now you have thousands of antibodies at your disposal. This is huge. You don't need to change them, just buy them, use them. We didn't have this before, so I get excited about this stuff. It's reliable, so it doesn't damage your tissue, and it's super fast. So let's have a look at how fast this thing is. You just saw that video go on. On the left-hand side, you see a nuclear stain. We're trying to bleach it with the light. You can see the decay curve in gray. On the right-hand side, you see the same stain, same conditions after we add Epic. The signal's done, like literally in 2 seconds. So I can label thousands of cells over millimeters of square footage, or square millimeters that would be, and yeah, the signal just goes off. So I can turn a light on and off at will. This is very good because it really streamlines my experimental design, and it allows me to move faster and also cleaner. So building on this in the last couple of months, and I think this is important for any end user, whether it's a core lab, academic, or pharma, or whatever, we built kits around these. So all of these antibodies are available, pre-kitted. There's instructions on how to use them. The colors are mixed and matched so that you can apply them in a panel. Our philosophy is only building small panels always. Don't build anything too large, and you can mix and match them as you need to. Probably the biggest advantage of this assay chemistry is it streamlines assay development. I think this is probably the most important thing also for a core lab. Again, having direct IF together with this fast signal removal technology allows you to tap into commercial antibody catalogs almost at will. You can go to Abcam, CST, wherever you want, buy these antibodies. and most of them are IF, immunofluorescence pre-validated, they will work on our system. We have a success rate of about north of 85% right now of hundreds and hundreds of antibodies that we've tested. This is better than any other technique that I know. And it includes antibodies against phosphorylation targets, which is notoriously hard to do in histology. I've spent years and years and years as a postdoc trying to label phosphorylation targets using standard IHC. It was always very hard. We've been able to do this, on the first go for many of the applications that we have. So it's agile, it's modular, and it really allows you to build assays as a quick clip. I think, again, for core lab, very important. One more thing about the assay, and then I'll briefly talk about an imaging feature. One of the things that is nice to do in a system, you do an experiment and you want to keep doing that experiment. It happens all the time in spatial biology. Here was a breast cancer tissue that we were looking at, and you can see in blue is cancer, is tumor tissue, and then you have this beautiful rainbow of immune cells around. It's probably like 10,000 immune cells. But they're just sitting around, they're hovering around, we don't know why. So this was the first iteration of our experiment. We put something like 20 antibodies on here. The question was very simple, immune profile this tumor. That's what we did. And then the researcher looked at this together with us, and the question was, why aren't these immune cells doing what's called infiltration? They should be going in there attacking this tumor. It's what we hope to do, especially when we give a drug. Well, we took that sample back out of the fridge, weeks later, put it back on the scope registers it, and then we could just follow up with additional stains. And that's the beauty of the system. Once your sample is locked into the flow chamber, you can probably go on and on and on, not probably, we've done this up to about 6, 7, 8 months, continuously reinterrogated the same tissue with additional antibodies. So yeah, this is how you get to 100 antibodies, and our philosophy is you shouldn't need to stain anything with 100 antibodies if you don't know what you're dealing with. If you're in a core lab, this is a real problem. Oftentimes, garbage comes in, and your data that come out are garbage. So you want to take as few antibodies as you want in the beginning, make sure your tissue is solid, and then you can continue. You can do that on that system. You don't commit to very much in the beginning. You can stain every tissue with just one antibody in the beginning if you want, and then keep going. So that's quite nice. It's quite flexible. So last thing I want to just briefly show you here is we generate a lot of data. Pictures are intensive. They take a lot of space. Our data are acquired at 180 nanometers per pixel. So a cell is no longer a cell. A cell is a complex architectural design that has axons, dendrites, has subcellular compartments. All of this is cataloged. That is data. We do this over a high dynamic range. That means we take several images per scene, in which we hope to capture low, moderate, and high expressing molecules. We do this every single frame, over several millimeters squared. And we do this over an imaging area that's 44 by 16 millimeters. So on a good day, on a single microscope, we could probably get up to 4 million cells at 182 nanometers per pixel with HDR. In reality, this is about 1 TB of data, which is a lot. And it's been a problem. It slows you down, costs money to store your data. So we recently revamped our software to keep all of these nice features. You can sort of see the quality of our data compared to a standard system. This is what happens when you photograph the same scene several times. You get higher sensitivity, higher dynamic range, and of course you do this on a large imaging window. But we managed to now compress our file size and reduce the acquisition in the time it takes to be exactly on par with our competitors. So this is hugely important. I'm no longer lugging around terabytes of data. I can actually, put it on a jump drive, and the people in my lab send it to me. This matters, especially if you're doing 50 experiments a week, 100 experiments a week. I have 12 of these machines in the lab. So this is really important. The end goal here really is, again, once, guys like these here come up with 12, 20, 30 protein targets, especially in a disease condition, my job is to see what these 12, 20, 30 protein targets do in a cohort of 100 patients, 200, sometimes even more, because that's how, druggable targets are truly verified. This is how translational biology happens. So that's what the CellScape is. This is really comes after discovery. It's once the hypothesis is made. This is built for that throughput, yet being very reliable, but also being very agile. And that's all I have to contribute. Thank you.
And you can just shout if you want, or use them.
I'm Ms. Brian from ARC Institute. So I'm more a genomics person, so maybe my question is a little bit naive. So my understanding of mass spec-based proteomics for the analysis part is you have kind of your protein sequences from the human reference, and then you match, the peptides that you sequence on the mass spec to them. That's how you know which gene you're sequencing. So if you're working with kind of a less model organism, what are the process if somebody comes to you is like, hey, I have this new organism that I want to do single cell proteomics, but let's say the genome is not that great, like microbial or something. What is the process of doing that?
If they have transcriptome data, you can build your protein database off of the translated transcriptome. And there's a number of nice software packages out there to do that. But you would need some sort of omic information to really do it in a robust manner.
So I have a follow-up, which is, okay, so there are, like people talk about, nanopore proteome, like sequencing. So I don't know what are your thoughts on, like, is mass spec-based single-cell proteome, like that's already there? We don't really need any more development there, or is there limitations to mass spec-based single-cell proteome? I know the other technology doesn't exist, sure.
Two mass spectrometers with exceptionally long conflict of interest with vendors got together and got a paper in Nature Methods that said that nanopore proteomics will never ever work in a million years. And so
I have that slide in some of my talks.
yeah, I think that it could, I do think that it will be interesting and can happen, and one protein at a time seems like it can be happening now. But we are pretty established here, right? In this space, it's
I guess my question is like, is there limitations to single cell proteomics done using the mass spec method that could potentially limit our ability to predict or understand biology?
I think the biggest draw for things like nanopore is the cost. So the mass specs are expensive, and I think that's the biggest draw.
Okay.
The downside with them, with different, any of the sequencing platforms, is if you just take what they're really good at, so quantifying or counting RNA or DNA, they don't produce as much information in terms of counts as what we already do with ion counting in the mass spec. So you can probably measure a protein in a nanopore, but you'd have to do it better than you can already do with RNA and DNA measurements. So there's two levels to that of, is it a theoretical and is it a practical?
Okay, thank you.
I'd also add in the copy number problem, right? That we can't amplify. We're limited to the raw amount of protein that's there. We all generate graphs for our material, where we try to guess how much protein we have that we're able to detect. If your protein's under 20,000 copies per cell, I'm not real confident that I can go after that with a global technique. I need to target it or something. So that's another limitation that
Low express.
Exactly, low express proteins. Okay. Don't forget that tomorrow at 10:45 we've got AI in proteomics, and it's not just like buzzword. It's actually like people that do AI in proteomics. I get some real stuff. So. Oliver, how can you get subcellular?
How can I get what?
Yeah, with your technique, are you getting subcellular?
Resolution?
Yeah, resolution, or is this?
Yeah, so we can differentiate compartments that you can stain well. Like I can see the nuclear envelope versus a nucleus. I think in our neuroscience applications, it's really, really important to be subcellular, because a lot of molecules will be expressed in an axon over, say, a dendrite or a cell body. And when you dissociate a cell, all this stuff breaks off and falls away. So yes, I wouldn't say we're super resolution imaging. That's all different linear problems. But yeah, 182 nanometers per pixel is subcellular. It's adequately subcellular for neuroscience applications and so on and so forth. Is it to study polarization of proteins in immune cells? Like, for example, T cells they'll have this molecule called PD-1, which is, a receptor. And the theory is that when one cell talks to another, the PD-1 localizes to one part of a membrane and somehow it changes the cell's behavior. You can sort of see it, but I wouldn't put my money on it. That's what super resolution imaging would be. So it's get more resolution. I talk about 20 nanometers or something like that, which is, quite a bit better. But yeah, we can do it.
Cool.
Hi, so Winston Temp, Johns Hopkins. So my question is, you guys introduced the idea of single cell RNA, and it's a very different assay. It's apples and oranges, right? But what, and then we were talking, you talked a little bit, Justin, about like the overlap between what you could see with single cell proteomics and single cell RNA. So what is it you can't see besides PTMs, which everybody loves? What is it you can't see with single cell RNA that you can see with single cell proteomics? Like, can you see, because proteins stick around for a while after the RNA is gone. And so is that something, have you looked at that, especially in your data set where you're looking at your plant root cells? Did you look to see, are there things that protein is showing up for that RNA isn't? And Ben, same question. Are you seeing stuff that you couldn't see with single cell RNA because RNA is just a readout of what's currently happening as opposed to what the cell might need, especially in cells like neurons, which you guys were talking about, that aren't mitotically active and might just have proteins hanging around for long periods of time?
Yeah, I think that's a proteomics room, so I think everyone's done the RNA protein correlation. But
Sure.
I mentioned one of the last slides, there's a nanopots, it's a custom platform for doing a lot of these assays. But it's a slide-based one developed at PNNL. And essentially they did a variant which is nanosplits. So they were able to, isolate the cells on a slide and sandwich them and essentially split the cell in half. And half of it they did sequencing on and half they did proteomics on.
Very nice.
And so they could actually address what is the correlation at an actual cell level, or is the correlation low before because it's just bulk heterogeneity.
Sure.
And here it's a similarly low correlation between transcript and protein. So I think it's not necessarily what you're measuring that's different, it's just...
But even RNA people know this, right? Because they're looking at translational efficiency and ribosome profiling, and people know that just because you have an RNA doesn't mean you have, one RNA is not equal to one protein. Everybody in this room, I'm sure, is very well aware of that. But especially at the scale that we're at for single cell proteomics now, that you guys have described pretty successfully, and I think it's pretty impressive, what can we now look at? Should we be looking for the proteins that pop up that you don't see with single cell RNA from a similar tissue? It won't be the same cells. What do you guys think we should be looking for or at? PTMs? What else?
I would, may I chime in here? So in spatial, over the last 3 or 4 years, multi-omics has been all the rage. And it was, we need to...
Hold on, can you define multi-omics? Because it means different things to different people.
Yeah, so to me it means simultaneously in the same section mapping an mRNA and a protein readout.
Fair enough, okay.
So we have a lot of data nowadays where we have done these experiments. Of course not. Well, we do have a whole transcriptome plus about 250 to 300 proteins.
Sure.
So that's
Nanostring
achievable today.
You guys bought Nanostring, right? That does that kind of stuff.
So that's enough to give you proxies to say whether or not these two readouts correlate. And it was always the assumption, especially for platform builders like us, we were always told, well, if you see this RNA, you have to see this protein, because they're related to one another. Turns out it's garbage. It's not how it works.
Sure.
It's completely different. So my answer to your question is, we should look at it all over again, not everything, because it's a completely different state of affairs. And yeah, PTMs, and then the 15 variants that every protein has, but I'm not convinced that single cell RNA-seq answered all of these questions yet.
It certainly didn't.
That's a different school of thought, and what these guys are doing is rewind 20 years, but a different substrate.
I wish you had reviewed the grant Ben and I wrote.
That's just what you said.
And Winston, I have an example in the KRAS inhibitors stuff that, when you look at the single-cell-seq, you look at the proteomics, the drug inhibits the active form of the protein. And so what we see if we look at the same time points with the single-cell-seq and we look at the single-cell proteomics, we see that some of these cells are trying, that their mechanism is trying to make lots and lots of the KRAS transcript, right? And so in culture, that doesn't work very well, right? Because I can just dump in more inhibitor, but biologically, you can't have it. So we start to see it's really important for that drug that if we took it to the pathway level, at the single-cell-seq level, single-cell proteomics level, we inhibited KRAS, we inhibited that KRAS pathway, all those normal things. But then when you looked at the two data sets beside each other, you see this really interesting thing of the cells that are just trying to overwhelm the pathway that we didn't see in the proteomics data because
So, as with all things, it's not one, it's we got to look at more facets, and so that's part of the power here.
Yeah.
Cool,
No,
great.
Thanks.
Thank you.
Yeah. And we do have lots of things where it's like, that protein's only at 10,000 copies, I didn't see it, but it has 8 transcripts, right? And that's enough for me to see it, right? So
PCR is beautiful.
yeah.
It helps us a lot.
Yeah. Cool. Anything else?
Just out of curiosity, how big is the transcriptome of the plant that you study?
Not changes, so 30,000 genes and then,
So it's more than human.
I think in a given cell, there's maybe 16 to 20,000 expressed.
And when it looks like you're doing what a single cell sequencing person would do. You use some k-means clustering or something, you root down a bunch of phenotypes. And assuming you take all 30,000 genes, what's the maximum number of phenotypes that are real that you've ever observed? Is it 12, 20, or is it really 100?
I don't know, I haven't thought about it.
Okay.
Yeah. I mean, you can mutate things, that's where usually are, and you see lots of different phenotypes.
Obviously, it depends on where you cut the plant. It's the root versus. Okay.
All right. Well, thank you, everyone. Oh, wait, no, wait, Sebastian. Sorry. I thought you were leaving. I thought you were starting to.
Yeah, so like I wanted to know like what's your sense of the current state of like reproducibility between batches in that specific technique? Because that's, in my experience, has been one of the places where analytical techniques start breaking apart. It works gorgeously in that batch that you made that day, but if you make one month from now, like the data does not look similar at all, right? Like what is your sense of that?
Question. So the first cell that I got to work right, mostly because I was teaching myself to grow cells at Hopkins during the pandemic, so I could do single cell proteomics, was H358, and I've used that cell line ever since because with one exception, I've been able to teach anybody to grow that cell line. And so that's I've been using that as a control, with the KRAS inhibitor. It's kind of and so while the protein in general changed, it's not a fantastic reproducibility as the technology's changed. Like as we've moved up through instruments, you can still see that we're inhibiting the pathway. But that's a great question. I went to a really scary FACS talk today, and I just found out that some of the FACS things sort at 2 psi, and some do it at 400 psi and blast them through a laser or something, right? And I literally do not know when I drop off the cells at core A versus core B what cell ends. So I think that those are fundamental questions we have to answer, right? Like how does that change now that I'm at Pitt and their sorters are not from the 90s, like they're modern things. I don't know how that's going to work, right? So that's a great question. I don't know if we've got a lot of data on HeLa cells. But all right. Thanks. Well, thank you, everybody.
Thank you.
That was so cool.
Likewise. It's fun to, I've always given the talk to biologists. So it's good.
Oh, yeah. That's
It's fun to mix it up.
those images are gorgeous.
Mal, fly back early.
Oh, hey. Well,
Good to see you.
Good to see
Great
you.
talk.
So this is
Justin,
all-
great talk.
Thank you, thank you. Now, yours is all 2D, right?
I mean, as well.
Yeah, we're working on 3D applications, but 2D is
yeah.
that's the main machine right now. Yeah. But yeah, that's the next thing, doing it in blocks.
Okay.
Yeah, but it's laborious enterprise.
That's cool.
Hey, you want to come down here? makes life